So yeah, I'm going to talk about environmental DNA today, which is something that you probably have all come across. It's an absolute pleasure to be in a room packed full of so much expertise on salmon. Some of the data sets that we've seen have been really incredible. This count data is fantastic, and actually, we don't have that kind of data for so many other species. But for the other fish, I'm just going to agree here. If we could play the video.
I know the human being and fish can coexist peacefully.
Now, I totally agree with George W. Bush. Thank you for your service, Mr. President. But we need all the tools that we can get to be able to create a coexistent future for freshwater fish. We know that they are beleaguered. A third of them are threatened globally with extinction in freshwaters. And, you know, migration being big, these are all factors that count against you. So surveying fish is inherently challenging. We know that. It requires our teams of experts in the field for extended periods of time. We're hoping that in that short time frame they're able to get representative data on what's living in the rivers. I think this example on the right really demonstrates that. We have a salmon par on the left and a brown trout par on the right. I hope I got that the right way around. But anyway, it just really highlights how difficult it is to distinguish between some species in the field, especially after a day's, soggy day's surveying. If you're a Chinese supermarket, that distinction doesn't really matter. But for us, it really does. And this was the only salmon par encountered in that situation. A really important data point that could easily be missed. So at Nature Metrics, we do things differently. Rather than relying on the two components of finding the animal and then identifying it, we use environmental DNA to detect the species. Environmental DNA lives in the environment for anything from 2 hours to 2 days, depending on temperature and sunlight, both of which degrade DNA faster. So for some periods of Iceland, you know, maybe it's a little bit longer than 2 days. And water really lends itself very nicely to eDNA collection. We're able to concentrate it just by filtering it, essentially. And that means that unlike kind of on soils or anything else, we can really, we can isolate this DNA from the environment. So at Nature Metrics, we essentially send out our kits all over the world. They look like this. And with minimal training, people are able to collect the environmental DNA. They fill up their water through the syringe, wearing gloves, of course, and then they connect the filter to it, and they push the water through the filter as much as they can. The filter will slowly start to kind of clog up with particulate matter, sand, mud, but also that environmental DNA. You then inject a buffer into the filter using this. Sorry, this is very much like a sales pitch, kind of is. And essentially, once you've injected the buffer in there, that stops the DNA from degrading. You seal it off and you send it back to our lab in Guildford, and that's where the magic really starts to happen. So essentially, we extract the DNA from the filter, but also removing it from all that particulate matter. We amplify the group of species of interest. This is metabarcoding, so we're not looking at a single species. We can, but it's much better to kind of get more bang for your buck. And we then sequence that data and then match up the data, the sequences that we found, to our online database. It's essentially like forensics at a crime scene. Exactly the same process. Instead of looking for murderers and rapists or what have you, we're actually looking for our species and kind of linking them to that scene. Okay, so don't need to talk about that. This is what the data looks like. So I want you to imagine that each of these columns is where's my trusty laser? Each of these columns is a kit that's been used, and each row is taxa. You can see that the salmon is ominously not there, but that's because it's a heavily impacted stream in England. But our founder, Kat, who is a DNA expert, collected the last 2 samples here, and you can see that she broadly got the same results as 8 first-time users of the kits who are citizen scientists. So, you know, there's a really objective, standardized way for people who, you know, don't have all the expertise that we have, that are able to sort of collect pretty good data. The bubbles are representative of how much data has come from that taxa. So you can see that roach, rutilus, and perch zander were the biggest contributors, really, with a bit of human DNA at the bottom. So this was using our vertebrate primer. Okay, I really like this example. So we're going international now. We're going to go to the mega-diverse Peruvian Amazon. WWF asked us if we could, do eDNA sampling of an area of jungle for these two species, the pink river dolphin and the Amazon manatee. These are the big, squishy kind of animals that get the donations rolling in from the public. So, they want to know if they're there and if they can kind of, put that in their comms and try and get some funds that way. The area is vast, so 1,500 kilometers of river, 250,000 square kilometers of jungle. We found 40 points along this sort of river network, and we used 4 kits at each point, and volunteers collected the samples. They used our vertebrate marker. And as you can see, they got this really nice mapping. You can see that the pink river dolphin is fairly ubiquitous to the whole area, but the manatee is sort of concentrated in this area down in the bottom right. We got 4 out of 4 samples for most of the river dolphin, and 3s, 1s, 4s, but clustered in this area here. So that's really important data in terms of managing that species. They know that they can prioritize their efforts in that area for the conservation of the manatee. They can work with indigenous groups to reduce harmful fishing practices. So really spectacular. But even better is that they got data on 673 other species of vertebrates from various different groups, and that means that they can do this mapping exercise with each and every one of them. So, really powerful, massive amount of data produced. Knowing how to use DNA and get the most out of it is about kind of, designing your design, essentially. So, eDNA is washed off the landscape from the headwaters and emitted by the creatures living within the catchment from the headwaters all the way down. So if you're just doing a sort of what's in my patch, what's in my river, you can just sample the main stem of the river, and that will give you a good idea of what's out there. If we want to get a little bit more sort of, understand the longitudinal zonation, we can start to add more sampling points and, split up that main river. We can then, add more points to look at individual tributaries. And then we can go for this like really high resolution species mapping approach, which can sort of highlight biodiversity hotspots, and also perhaps like tributaries that are impacted and not as diverse as we would expect. And then we can start to link that to sort of landscape pressures and other impacts. This approach was really pioneered by Luca Carrera and Florian Automat, who are some Swiss academics. And you can see how, on the bottom right, how they've kind of split up this confluence, split up this catchment by selecting the confluences at which to monitor. And this was actually for invertebrates, so looking at freshwater insects, they can create a kind of heat map of the areas in which the insects are found in the river using eDNA. We've got some really exciting projects. We're working with Lorraine and her team on the River Dee and Don. I think this is the Don, I hope so. But essentially, we are sampling from the sea at the estuary on the right, near Aberdeen, all the way up the main stem of the river, and also looking at a few tributaries too. They are sampling in spring, so they're getting the spring salmon run that we all heard about yesterday, and also in autumn. So they're going to get the pink salmon run, which we heard the minister talking about yesterday. We don't really know what the impacts of the pink salmon are, or how far they're going upriver. So the results from that will be really interesting. Lorraine and her team have also done their homework. They're doing using our invertebrate marker alongside the fish marker, so they can pair that data together, and they can actually start to sort of see build up the faunal community for those systems, which will be really interesting. We are developing a digital dashboard. This is going to make bringing biodiversity data into the boardroom much easier and make it accessible to everyone. Biodiversity is the new carbon, but we don't really know how to quantify or measure it. And we also know that carbon is kind of the same wherever you are in the world, but we know that biodiversity is inherently different. Some biodiversity, salmon is better than others, that sort of thing. So it'd be really interesting to see how this dashboard plays out. We work with a range of sectors around the world, anything from academics to NGOs, and we are producing vast amounts of data on an annual sort of basis. And as the uptake increases using environmental DNA, there's almost like we don't really know where to put this. We're getting it from, as you can see, we're operating in quite a few countries. I think this slide is slightly outdated. You know, I've started working on some projects in Oman recently, so we're constantly adding to this global portfolio. Which brings me lastly onto our next project, eBioAtlas, which is a collaboration with the IUCN, where we're essentially building a biodiversity layer for the world using environmental DNA. We can try and fill the Wallacean shortfall, where species are, what their ranges are, and where they're found, but also allow for sort of desktop analysis to assist with conservation strategies, planning for infrastructure, and all that sort of thing. Thank you so much for listening to my talk, and I'll be happy to answer any questions, not now, but come and find me around lunchtime. Thank you.